Welcome back to Bioinformatics. Today we are going to know what is pairwise alignment and multiple sequence alignment. So what is a sequence alignment in bioinformatics? It is used to compare the DNA, RNA and protein sequence to identify the similarities. Then pairwise alignment, it compares two sequences to detect the homology, and with multiple sequence alignment, we can align three or more sequences to retrieve, conserve regions and evolutionary relationship for constructing an phylogenetic tree. So what is first the pairwise sequence alignment? Pairwise method compare exactly two sequences to find the high scoring arrangement of matches, mismatches and gaps. And global alignment align the sequence across the entire length. It is best for sequencing of similar length that are large homologous like the Needleman bunch. It used the dynamic programming. Then local alignment, it find the high similarity subregions within longer sequence, and the idea is for comparing a strong query sequence to as much longer genome or looking for a conserved function domain. Example Smith-Waterman. The heuristic tools, it is a method like BLAST, bypass strict dynamic programming to quickly scan large database. It is used as a basic database search, mapping read to a reference genome and identifying a specific mutant. Now, multiple sequence alignment, it aligns three or more sequences simultaneously to identify the conserved pattern across a whole family of sequence. The progressive alignment, it builds a guide tree by calculating the pairwise distance between all the sequence, then progressively align the most similar sequence like cluster omega or MAFT. Then iterative alignment, it aligns the sequence initially and then repeatedly realigns subgroup to refine the alignment and minimize the error made in earlier stage like muscle and prank. Then uses, it is used to construct the phylogenetic tree, predicting protein structure, tertiary structure and identify active site. What is the key difference between pairwise sequence alignment and multiple sequence alignment? The number of sequences is exactly 2 in pairwise sequence alignment, whereas in multiple sequence alignment 3 or more are there sequences. Then computational complexity is lower, high and efficient in the pairwise, whereas high and computative expressive in the multiple sequence alignment. The primary output of pairwise sequence alignment is identity similarity scores or aligned pair, whereas for multiple sequence alignment it is conserve motif consensus sequences phylogenies. Then best it is best used for the pairwise sequence alignment is best used for fast database search and query annotations, whereas the multiple sequence alignment it is best used for evolutionary biology and structural analysis. Now, scoring and matrix. Both alignment types rely on the scoring system to evaluate the accuracy. Positive score reward matches, while negative score penalize mismatch and the insertion and extension of the gaps. The nucleotide offers score with a simple identity matrix, example +1 for the match and -1 for mismatch. And the protein, the score used the substitute matrix like PAM and BLOSSOM, example BLOSSOM 62, which account for the likelihood of specific amino acid substitution based on the physiochemical properties. Here we can see the multiple sequence alignment by Ka. We just have to give the three FASTA formats, more than three FASTA formats for multiple sequence alignment. For two FASTA formats, we can use the pairwise sequence alignment. So here we will paste some of the FASTA sequences, and after that we will execute multiple alignment. See a ClustelW. It is widely used in bioinformatics tool used to perform multiple sequence alignment for DNA, RNA and protein sequence. So it helps in aligning the most of the similar sequence first and progressively adding more distinct sequence to build the comprehensive alignment. How does it works? The process unfolds the three major stages. It ensures the high quality biological accuracy alignments. So here in multiple sequence alignment we have to paste a FASTA format here. Total 3 format we are going to paste. For example, I am taking a protein sequence. and pasting it. Now let's compare and execute the three sequences. Now similar way I am going to copy and paste three sequences. After executing we get a result here. There is a asterisk colon period and blank space. So the asterisk, what does it indicates? The position that have a single fully conserved residue. The colon, it indicates highly conserved position with strong similar properties, example the conserving amino acid mutation. Then period is a dot that indicates the weakly similar properties, and blank space, it indicates the lower similarities or introducing the gap to optimize the sequence alignment. Now we have to select the tree menu, and after that we have to select the fast tree. Execute. Now on clicking this we will construct a phylogenetic tree. So in this diagram we can see the output web page of phylogenetic analysis pipeline generated by software ET-E3, that is evolutionary genealogy tools hosted on the genome net. So it presents a reconstructed evolutionary tree showing the relationship between the three genetic or protein sequence. Here we can see the protein sequences, and here it is breaking down of the key section. The workflow method, it shows the default fast tree pipeline, that is default as we selected. The alignment and construction is seen. It is used the E3 build function to automatically construct the tree. The tree builder specific evolutionary tree was constructed using the fast tree, and the node value, that is sh, like local support value, the number at the branch junction, that are the node that represent the sh like local support value, that is Shimodarial Hezgawa local support value, something like this name is there, which indicate how statistical confidence the pipeline is in the specific branching split. So here the .nwk, this is NIC format. The text-based file contains the mathematical architecture of both the unrooted and midpoint rooted tree, and WFA or FASTA, the multi-sequence alignment text that file used as a foundation to compute the tree. The phylogenetic diagram, the scale bar, the horizontal griddle line tracking from 0 to 1.4 represent evolutionary distance, typically measured in genetic mutation or substitution per sites. The sequence, leaf, tip, the three specific sequences are being compared. NP, that is top branch, then SP here, the human one, that is a middle branch, human insulin like growth factor 1 receptor, and KI is the bottom branch. We can see the three sequences, and the branch length, the long horizontal line extending the top sequence, that is NP. We can see here the number after that. It shows that it has an accumulated significantly more evolutionary changes or mutation compared to the other two. And there's a clade, that is a grouping, the human sequence, that is IGF1R human, and the third sequence KAI. It shares a much closer and more recent common ancestor with each other than they do with the top sequence. So the node joining them display a confidence support of value of 0.5, that is 100. So for knowing such concepts, follow Bioinformatics. Thanks for watching. Do like, share and subscribe with your friends.